hear me? Yes. So first of all, thanks to the organizer for giving me the opportunity to present my work here. I work with Vera Gorbunova in Rochester. And today I want to talk in general, we already saw about axolotl, but in general, what is regulating the dynamic of protein synthesis across mammalian species and what are the implications for the mammalian lifespan, basically. We know that aging actually alters all the patterns of the gene expression program, from genes to protein. And we are starting to understand how aging alters the general structure of the transcriptome with, for example, loss of expression of long genes, change in polymerase speed, and also stalling. Here, my work is also contributing to understand how actually the second layer of information from mRNA to protein is actually affected. And here we are looking into phenomena of translation posing that seems to be related to changes and in protein homeostasis. What I think is interesting to understand, especially for the general biology of aging, is how this general system actually evolves across mammals. Because only in this way we will really be able to understand which are actually the mechanisms that drive longevity and lifespan across different organisms. So this is exactly the objective of our lab, and we collected actually an extensive data set of mRNA and protein characterization from 6 different tissues across a large variety of mammalian species with different lifespan to understand how the relationship between the layers, mRNA and protein, evolves and together with lifespan. To do this kind of analysis is really important to really have the correct genomic tools, and here we are collaborating with the Comparative Genomic Lab, applying their software to really understand the evolution of specific genes via orthology mapping. And here, once we set up all this pipeline, we can basically start to explore how protein levels evolve. Here is a simple example, really simple example. On the x-axis, you have the different species, and on the y-axis, abundance of both protein and mRNA. And in this case, you can clearly see that, at least for this specific case, the protein level across species are actually modulated mostly transcriptionally. This means that if a specific organism needs to produce a specific amount of protein, this might specifically happen just by modulating the abundance of the corresponding mRNA. We can, of course, go and look what are these functions, how this function evolves, and we found that the transcriptional response is actually related to a lot of parts and pathways that are actually very important in regulating aging and lifespan. We have a lot of mitochondrial component, but also extracellular matrix component and so on. This indicates different modalities of regulation for these specific pathways. We were actually very interested in specific categories of regulation, which is actually the opposite, virtually the opposite, where protein and mRNA level basically evolve mostly independently, or in some cases even the opposite way. We call these cases post-transcriptional regulation because it really indicates that the modulation of the protein level is happening independently of the transcriptome. And what are these functions? Here again, very interesting, we found component in this case of the protein synthesis machinery such as ribosomes, and also importantly component related to the chromatin complex and DNA repair. These are again very important pathways that are known to modulate aging and lifespan. And of course, what we want to understand is actually what is driving this changing, how these post-transcriptional proteins are modulated. And here we are doing a great collaboration with Andreas Beyer in CICAD in Cologne, where we are basically looking at the complete structure of the transcriptome across different species to really understand how, for example, mRNA metabolism contribute to protein level. And here I give a simple example of what I'm talking about. We can, for example, calculate measure of splicing efficiency by comparing from RNA-seq dataset reads that map between different regions of the gene, in this specific case exon intron, and reads that are mapped between different exons to really give a matrix of how, for example, a specific genes get spliced, for example. And then we can do basically the same approach. We can take our measure of splicing efficiency and we can simply correlate by correcting for the phylogeny that persists between the organisms and really understand, okay, what are the proteins that are actually evolving with the splicing efficiency. And here, interesting case again, we found that those proteins are again related somehow to this post-transcriptional mechanism that I just talked about, especially for what regards the processing of ribosomal RNA. This is very interesting, but how this is related to lifespan of the organism? Well, if we look at the proteins that are correlated with maximum lifespan, whose abundance is correlated with maximum lifespan across organisms, we actually find that a large proportion of this protein is actually modulated independently of their transcript level. This means that these different species are actually acting in between to modulate protein abundance. And here I really want to give you a strong example of this, because the cytoplasmic ribosome seems to be one of the most strongest signals that is coming out from these comparative studies. We know that ribosomes are central machine in all the metabolism they produce, and they are the main actor of protein synthesis and are also the final effector of the gene expression program. And we have this characteristic regulation where we have a decrease in ribosomal protein abundance, which is strangely followed by an increase of their transcript level. And indeed, this is also true because when we look at the correlation of mRNA and protein for cytoplasmic ribosomes, we see that this correlation is virtually zero. This means that we cannot, the ribosomal protein, and in general probably protein biosynthesis, is evolving independently of the mRNA. Now we can ask, we have our measure, for example, of splicing efficiency, we can overlay, and we can see that when we consider the opposite splicing, we actually get a slightly better prediction. This might indicate that, in this case, different organisms are modulating ribosomal protein level via splicing. Of course, mRNA biology is not the only key player in modulating protein level. We have also, of course, protein turnover. And this is a very important mechanism that regulates protein across different organisms. And here we have the luck to have Sina Gamagami in our university in Rochester, who is a great expert of protein turnover. And he already characterized actually protein turnover data across a large variety of organisms. And he found actually an interesting negative correlation between the amount of protein turnover and the lifespan of the different organisms. What we can do is that we can leverage this data that were already produced and try to understand how is protein turnover affecting, for example, ribosome dynamic. And when we put this into context, we see that actually when we overlay protein turnover data, we can actually fairly well predict ribosomal protein abundance across species. This really suggests that different species are modulating ribosome abundance by controlling actually their synthesis and protein turnover. This is not the only convergence that we see happening at the level of the ribosomes. We find a very interesting association, and in particular, this interesting association between the length of ribosomal genes and organism maximum lifespan, to the point that you can really fairly well predict how an organism will live just by looking at the total genes length of ribosomes. And this seems to be something that is clearly very well related just to lifespan, because when we, for example, look at other characteristics that we know are connected with lifespan, such as body mass, we find no significant association, basically. We are actually trying to understand how these two layers of regulation might actually interconnect, and we are doing work in this sense. So what we see is that there is a decrease in ribosomal level that is happening post-transcriptionally, but does this as an effect on protein synthesis? So we know that protein synthesis is generally detrimental for aging and lifespan. We know that multiple drugs that we have also shown today, observed today, act in the end on the protein synthesis pathway by lowering it. So a question will be, do these long-lived organisms actually reduce their protein synthesis level by controlling the ribosome abundance? And to do so, what I'm doing is just basically measuring protein synthesis rate in different cell lines from these different species to understand how protein synthesis evolve in relation with lifespan. We actually, not surprisingly, kind of expectedly, we found a negative correlation between the rate of protein synthesis and organism maximum lifespan. And interesting, this relationship even gets stronger when we correct, for example, for animal body mass. This might suggest really that protein synthesis might actually be a pathway that could control both of these processes, both body mass and lifespan. So this is for the general mechanism of how protein synthesis might actually evolve across organisms. There is another interesting observation that I did during my PhD study, where actually very similar phenomena are actually happening also in the aging process. So there seems to be, here we are looking at the killifish aging brain, and here in red again we have protein, and in blue we have transcript level, and we see virtually the same observed regulation, a decrease in ribosomal protein level, which is followed by an increase of their transcript. This might suggest that protein synthesis, the regulation of protein synthesis might not be so easily associated with lifespan. There are complex mechanisms that are linking these two processes together. And what we are proposing in this work is that actually there are alterations in the translation landscape. We did the translation profiling with the ribosome profiling, and we see specifically increased translation posing, so ribosome posing at specific codon that are encoded for basic amino acids such as lysine and arginine, which are actually very enriched in ribosomes and RNA binding protein in general. So to give a small summary, so we are kind of in a scenario where we have for an increased ribosome synthesis, and actually ribosome biogenesis, we can say, leads to increased ribosome level, and in the end, increased protein synthesis with my lead in actually in a live fast, die young state. So this short-lived organism might actually burn through this ribosome biogenesis pathway and live very an accelerated life, while on the opposite, reducing ribosomal synthesis and turnover might lead to a reduced ribosome level, reduced protein synthesis, and this might actually lead to extended lifespan. And we're also actually starting to see that several changes in gene properties and characteristics are actually linking and pointing to this specific adaptation of these long-lived organisms in their protein synthesis machinery. With this, I want really to thank, first of all, Vera and Andreas, who are also here at this conference, that really supported me when I came to them and I told them that I wanted to work on this project. And of course, also all the people in Vera's lab, especially Greg and Cheyenne, who are helping me with all the project, and all the collaborators that are also participating in this big endeavor. And I'm ready to take questions.
Great talk. Do we have any questions?
Awesome talk. I was curious about, are there known regulators of ribosomal protein turnover? Does this happen at the transcriptional level? Are there known regulatory mechanisms? Or is this more a genome intrinsic thing?
Yes, so this is interesting. So there are several, so in general, the process of ribosome biogenesis is very complex, especially in mammals, and not everything is understood. So what we know is that usually some of this regulation, especially maybe across tissue, because this was measured before, they see that it happens mostly at the post-transcriptional level. But of course, when you produce ribosome, you need to produce kind of in a concerted way. You want them to be a highly regulated process. What I know is that at the level of transcription of ribosomal protein subunit, not so much is actually known about how these processes are regulated. What we see is that across species, let's say the most easiest and convenient way to change your ribosome level might actually be just changing your translation rate, or how much protein you produce of this ribosome. We don't see this happen so much in other different manner. As I showed, the correlation with mRNA is basically 0, more or less. So, yeah. Thank you.
Yeah, here, the question is here, from here.
Ah, okay.
Yeah, it also in our analysis also, like ribosomes appear all the time, and once we study longevity, and there's a paradox that gene expression of many ribosomal genes increases with the increase in lifespan, whereas the translation kind of decreases. What's your kind of explanation?
This is probably the most interesting part about this. So what I think is that actually this increase in transcript abundance which we see could actually not be coming from novel transcription. What it might actually be is that, what I suppose, but I'm still trying to look into a bit more carefully into the data, but I think there are changes probably in the turnover of this mRNA that might lead to this kind of apparent discrepancies. It will be interesting, of course, to link these changes in gene structure also to how this is reflected in the structure of the, let's say, of the transcription of these subunits. And it's something that is for sure very interesting, and I'm really trying to understand what is going on in this sense.
Last question.
Quick question over here. Of course, I'm interested in the extra matrix, and I like to correlation mRNA to the protein. Is that tissue specific? Do you see it in all the tissues?
So there is a slide probably where I really show it by tissue. Where is it? So here. So seems to be more or less, so there are some tissue here, I didn't put the label, maybe I need to double check better, but seems to be fairly, let's say, conserved, I would say. There are some difference, of course, across tissues, and this might be, of course, reflect really the biology of this. Of course, probably, especially for extracellular matrix, it might not just be a transcriptional response, might actually be also kind of a mix of the two. Ribosome, for sure, is the strongest signal that comes out from this data, but there are also convergence on other different pathways, and actually protein turnover of extracellular matrix might actually be probably also a strong component that is contributing to the level of this protein.
All right. Thank you so much. Great talk.
Thank you.